Okay, welcome back. This is the last part of Chapter 12, and I'm going to talk now about sex determination, and this is really tied in with sort of the last exception to Mendelian genetics that I wanted to go over, which is what's called sex linkage, sex chromosome linkage. And so I want to introduce this idea of sex chromosomes. You may be aware of this already, but in many animals, right, sex is determined by either the presence or absence of specific sex chromosomes. And sex chromosomes are distinguished between non-sex chromosomes, which are called autosomes. So in the human genome, right, we have a single pair of sex chromosomes. All the others are autosomes. And you're probably familiar with sex determination in mammals, humans included. Right? Females are considered homogametic, meaning they have two of the same copies of the particular sex chromosome, right? They're XX, whereas males are heterogametic, meaning they have one X sex chromosome and one Y. So XX female, XY male. The Y chromosome is much smaller than the X chromosome, so actually have less genetic information than females do. Might explain a few things. But I want to point out this is not the only mechanism in the natural world. So here I'm going to go through this table kind of backwards. You don't have to memorize these different mechanisms for the test, but I just want you to be aware, right? So we just talked about mammals, right? Males XY, females XX. In birds, it's basically just the opposite scenario. So instead of XY, they're WZ, are the sex chromosomes, but males are homogametic, WW, and it's the females that are heterogametic, WZ. In fruit flies, honestly, in fruit flies, it tends to work out to be basically the same as in humans, but it's actually technically sex being determined by the ratio of sex chromosomes to autosomes. And then in bees, it's a whole nother ball of wax, so to speak. You've got males are diploid, females are haploid. If you remember those terms, diploid means you've got two copies of every chromosome. Haploid, just one copy. So there's like a dramatic difference in chromosome number between male and female bees. And we talked in previous chapter about this idea of non-disjunction, right? Chromosomes not separating correctly, or leading to things like Down syndrome. And I mentioned the fact that non-disjunction is not well tolerated for most chromosomes in humans. You know, one exception to that is like the smaller autosomes, chromosome 21 or 22. You can have trisomies or monosomies there. The other exception are the sex chromosomes. So humans can tolerate very well sort of unusual numbers of sex chromosomes. And there's some pretty relatively common syndromes that result. For example, Turner syndrome would be an individual that just has a single X. So they're going to be phenotypically female, but typically sterile. Klinefelter syndrome, got two X's and a Y. Those individuals are going to be phenotypically male for reasons I'll talk about here in a minute, but will also be sterile. And there are some sort of subtle phenotypic effects, but male for male biologically speaking. And so interestingly, in humans, we talk about sex determination. It really boils down to just one single gene, this SRY gene, sex determining region, that is found on the Y chromosome. And the bottom line is, if you have the SRY gene, you will go through embryonic development as a male, right? You'll develop testes, whereas if you lack the SRY gene, you don't develop testes and you develop ovaries instead. Right? So if we go back to those syndromes, right, that makes sense. XO, we don't have the Y chromosome, we don't have the SRY gene, so we're going to be phenotypically female. Klinefelter syndrome, even though we have two X's, which normally you'd associate with female, you have the Y chromosome, you have the SRY gene, so you're going to develop as a male. So kind of crazy, all the differences between the sexes really all boils down to that one particular gene being present or absent early in embryonic development. And so to tie this back in with these exceptions to Mendelian genetics, if you have genes controlling traits that you're interested in that are located on the sex chromosomes, right, that will give you some interesting inheritance patterns. And so I mentioned eye color in fruit flies earlier. Typically, fruit flies have red eyes, but through looking at some mutants that had white eyes and the inheritance pattern, researchers, again, I think this was from Thomas Hunt Morgan's group, discovered that, oh, the eye color gene is what we would call a sex-linked trait, meaning that gene is located on a sex chromosome, and so we get these, again, these kind of unusual patterns of inheritance. So I don't want to get too deep into it, but the essence of it is if you see traits that appear a lot more often in males than in females, like the eye color gene in this particular example, that suggests that that gene is sex-linked and is probably located on the X chromosome. And the reason you're seeing it more often in males, in this case white eye color is recessive, but remember males, they're going to be XY, so they only have the one version of that eye color gene, right? There's no second X chromosome that might have a dominant allele that could mask that recessive white eye gene, so it's going to appear a lot more often in males. Okay, so anytime you see something like that, that would be a hint, maybe there's some sex linkage going on with this particular trait, and that happens pretty regularly. So hopefully you're getting the idea, right? Even though Mendelian genetics and inheritance is pretty nice and tidy, things can and do get more complicated, and I think arguably more interesting, right? I think some of these exceptions are what make genetics so fun and challenging to think about. Yeah, so this term carrier, we've seen this before, right? So these are going to be people that really in any instance, whether you're talking sex linkage or not, but an organism that carries a recessive allele and could pass it on, but doesn't actually have that particular phenotype. All right, I said that was the last example. I've got one more for you here, and this one's really cool. So you might remember we talked about the fact that chloroplasts and mitochondria were certainly at one point free-living bacteria that are now organelles inside of eukaryotic cells, but they are the only two organelles that have their own DNA, right, aside from the nuclear DNA. And what's interesting is because sperm have so little cytoplasm, and that's where you find mitochondria, whereas eggs have lots of cytoplasm, you basically inherit all of your mitochondria from your mother, right? They're present in the egg, not so much in the sperm. So that mitochondrial DNA, which is actually very important, right? Mitochondria carry out these, key energy reactions in your cells, right? All of that DNA, genetics, that biochemistry is linked to your mother. And in fact, you can really think of it as there is an unbroken chain of mitochondria from you to your mom, to your mom's mom, right? All the way back to, wherever your family line started, right? You've got those mitochondria being handed down directly, mother to child, mother to child. And I've got this funny little video. This guy wrote a song about the importance of maternal inheritance. So he wrote it for his mom for Mother's Day, but it's pretty clever. And I think he was a bio major, so he's got a fair amount of solid biology in there too. But I'll try to remember to link to that on YouTube so you can check it out. But yeah, there's this phenomenon called maternal inheritance. All right, then the very last couple slides in this chapter are talking about prokaryotes, bacteria. How do they transmit their genes, right? I told you in earlier chapters, oh, these organisms reproduce asexually through mitosis or binary fission. In the case of bacteria, they're just creating clones of each other. But of course, that's not always the case. Bacteria can evolve, their DNA can change through mutation, but also through some of these sort of mating type mechanisms that can occur, even though it's technically not mating. But in certain species, bacteria can form what are called sex pili, which is basically like a little tube that connects the cytoplasm of the two bacteria, and DNA can actually be transferred through that conjugation tube, that sex pilus, from one bacterium to the next. I guess I sort of used the wrong terms there. So just to clarify, technically the sex pilus is really just the pili, these projections that initiate the contact that then leads to the conjugation tube, which is actually allowing the passage of DNA. But anyway, it could look something like this, where you have pretty significant chunks of DNA moving from one to another. Okay, via conjugation, bacteria are pretty good at incorporating new DNA into their chromosome, so you can actually get quite a bit of DNA transfer and shuffling of the genomes of these bacteria in that manner. Another way bacteria can share DNA, again, this kind of goes back to conjugation tubes here, is via what are called plasmids. Plasmids are these small circular pieces of DNA that you find in a lot of bacterial species. They're not associated with the bacterial chromosome, right, which is a big circular piece of DNA. These are sort of like extra little bits of DNA, and they can actually contain genes. For example, a lot of the genes for antibiotic resistance are found on these plasmids. And because these guys are small, they can very easily be transferred through these conjugation tubes if they form. And this is known to be one mechanism by which bacteria can pretty rapidly acquire antibiotic resistance, which is a big problem these days, as I'm sure you're aware. So anyway, that's it on chapter 12, and I will talk to you next time for chapter 13, which will dig into DNA and discovery of DNA as a genetic material, and how it